Hello and good afternoon. My name is Tim Poe. I'm Director of Telehealth with the UNC Cancer Network. Thank you so much for being here today, January 24th, 2018, for our Medical and Surgical Oncology Lecture. We're very glad to have you here. Just a few things I want to go over and then we'll get started with our lecture today. You can contact us by email, unccn@unc.edu. You can find us and contact us by telephone, and that's probably the best way to get a hold of us right away, especially if you're experiencing any technical difficulties. So if you're running into any problems at all, either with garbled audio or bad video, or can't see the slides, etc., please give us a call right now, 919-445-1000, and we'll do everything we can to help you before the presentation starts. We'll be using Poll Everywhere in just a few minutes. You can find us on the web at unccn.org. Find us on Facebook, Twitter, and YouTube as well. All right, we'll start off by using Poll Everywhere and asking you to go ahead and respond to a poll. This is anonymous. And the way you respond to this poll, you can either go to pollev.com/UNCCN on a web browser, and that includes a web browser on a smartphone, and you'll see the question there. An alternative is that you can use your phone, and you can text to the number 22333, the letters UNCCN. Just do that one time. You'll get a little message back that says join, and then you can respond to the poll. Good news about that is that once you've done that, then you can also ask questions in our Q&A session at the end of the presentation. So the question for today is, according to the journal Nature, a major cause of genetic heterogeneity in cancer is, and then you can pick one of the following, A, B, C, or D. If you think it's option A, subclonal uniformity, you'll go ahead and send in an A. If you think it's option B, identical gene expression, go ahead and send B. C is genomic instability, and then option D is genomic stability. So go ahead and take a shot at that. One of those four answers, go ahead and text that in after you've joined by texting 22333 with the letters UNCCN. All right. We're very happy to have our guest here today, and he is Dr. Jason Akulian. Dr. Akulian, welcome. Thank you so much for being here today.
Thank you.
Dr. Jason Akulian is an assistant professor of medicine and director of interventional pulmonology in the Division of Pulmonary Diseases and Critical Care Medicine. His clinical expertise is in the diagnosis of thoracic malignancy, as well as airway and pleural intervention for diagnosis, treatment, and palliation. His current role is in the Department of Medicine, is to build a world-class interventional pulmonology program by which the Department of Medicine will provide cutting-edge care to patients with malignant and benign causes of airway disease while teaching the next generation of physicians. Sound like very, very worthy goals. We're excited to hear about that. Let's take a look at that poll. We're running heavily in favor of option C, that genetic instability. Dr. Akulian, how are our guests doing today?
Looks like they're doing pretty well. That's absolutely what we're going to be talking about today.
Great. Well, thank you to all of you who responded to that poll. And again, if you have not yet joined, go ahead and join and be thinking about questions that you have for Dr. Akulian that you can share with us at the end of the presentation. I do have a disclosure statement to read. This activity is planned and implemented under the sole supervision of Thomas Shea, MD, the course director, in association with the Office of Continuing Professional Development. Thomas Shea, MD, consults for Spectrum Pharma and receives research support from Millennium, Otsuka, GSK, BMS, Novartis, and Seattle Genetics. CPD staff have no financial relationships with commercial interests. The speaker, Jason Akulian, MD, MPH, consults for Baron Medical, Fujifilm, at Boston Scientific, and Medtronic. All right. And with that out of the way, we'll turn it over to you. So here's the mechanism to advance the slides and the cursor. Lung cancer heterogeneity and its clinical applications.
All right. Thank you very much.
All right. Thank you, Dr. Akulian.
I would have one disclosure that was recent that I didn't get a chance to update was Becton Dickinson as well.
All right. Thank you very much.
So everyone's comfortable with it.
We're in the clear.
All right. Very good. Well, I want to thank the Lung Cancer Network, Lineberger, everyone who's here to show up and actually listen to me give a talk. and Tim for having me. Today we're going to talk about lung cancer heterogeneity and its clinical application. So what do we actually do with it? What is it as an idea? And then how have we actually harnessed it for a patient treatment standpoint? So again, related to this presentation, I have no disclosures, but you heard my disclosures earlier globally. So let's talk a little bit about heterogeneity and what that actually means, right? So heterogeneity as defined is just a variation of something within a whole. So cancer evolution, however, if we look at it, which is all part of this big picture, going back all the way more than 100 years ago, von Hausenmann actually was looking at these cells underneath a microscope and seeing abnormal mitosis or dividing of the cells in cancer cells, and theorized that and hypothesized that this may give rise to an asymmetric chromosome distribution. And I don't think he understood what that ultimately would mean, but he definitely understood that there was something going on at the genetic level or within the chromosomal level of a cancer cell that would ultimately cause it to become increasingly dysregulated. Jump forward more than 60 years, you have Huxley, who coined the term the genetic inhomogeneity of cancer, right? So again, looking at this heterogeneity or inhomogeneity, which is another way of saying the same thing. And again, he's asking, this is well before our time, when there were very limited laboratory techniques, but he was asking, saying it would be great interest to discover the extent of how much variance there actually is within tumors and how often it occurs. And then even he further theorized and how we might take advantage of that potentially. Right, so this again was predating chemotherapeutics as we know them today, certainly targeted therapies or immune therapies. But again, this idea that there was this dysregulation in chromosomal distribution and genetic distribution within cancer has been around for a long time, and how to deal with it is really where we're at now. So cancer unto itself is genetically unstable. It's part of the reason why it happens, why we actually get cancer is we have damage to our DNA through a series of processes, the multi-hit causation model, that ultimately result in a viable cell that then divides on its own, has lost its regulatory functions, and just divides out of control. Ultimately, those same genetic mechanisms, however, make the chromosome and gene array within the actual tumor cell unstable and gives rise to further heterogeneity. So you have this idea that there's an increased chance of daughter cell creation that is unlike the mother, right? So we think of mothers and daughters and sons and fathers as being similar to one another, but not exactly the same, right? We're not clones of one another. This leads to intra and intertumor cellular heterogeneity. So when you look at two tumors within a single person or even two cells within a single tumor, they're oftentimes very different. When you look at two tumors between two people or even two cells within two tumors between two people, they're also quite strikingly different. There appears to be this sort of optimal range of instability, meaning that if the cell is not too unstable, then you kind of do have this clonal distribution where you get a preponderance or a dominance of a single type of cell, and oftentimes those are dealt with through a number of different mechanisms that the body has to clean up cancer. I mean, if you think about why we have an immune system, it's not to fight bacteria and fungus originally. It was to clean up all of the cancer that we make on a daily basis in our bodies. That said, if the cancer cell is too unstable, then it'll actually sort of divide itself out of existence because none of the cells will ultimately become viable. So there's this sort of optimal range of instability where the cancer actually is able to divide unchecked and create subclones or even daughter cells or child cells of itself that are also viable, and they give rise to further subclones that have differing genetic viability. And this is part of this idea of how cancer then ultimately spreads and how it's treated, and these general ideas we're going to talk about. So as the tumors grow, again, they evolve because they're constantly dividing. The cells are dividing. They're unstable. Their genetic makeup is changing within the tumor and within tumors that they then metastasize. And so you have this increasing heterogeneity within the entire tumor body. Typically, you have about 1.6 to 9 mutations per cell division, right? So again, this is that sort of optimal range of instability. Less than that, the tumor is less viable. More than that, and again, the tumor is also less viable. And if you start to think about what this actually means, by the time we can actually see a tumor on radiography or actually physically see it on the patient, if it was a skin metastasis or a lymph node or something like that. We're talking about now millions and billions of cells at that point, and so they've now undergone multiple turnovers and mitoses, and now have millions of mutations that they've acquired, some of which have died off, so those cells ultimately are non-viable. But there's always going to be this selection pressure in one way or another that we're going to talk about in a moment. So some of the intrinsic sources of heterogeneity are the actual just genes themselves. So you have mismatch repair and microsatellite instability, so actual unstable regions of DNA due to methylation or demethylation or different folding structures that don't allow for the DNA itself to become stable. Mismatch repair, so we have a whole litany of ways that our body deals with incorrect base pairing, right? So we're constantly, again, making improper cells. Our bodies are designed to come along and fix those incorrections. It fixes things where they need to excise something that's wrong and put in something new to, again, repair the DNA so that we don't die or cause or create cancer. And then if you have damage to a base pair by, say, ultraviolet light or some kind of a carcinogen that you ingest or are exposed to, you have a way to then repair the DNA that has been damaged. Ultimately, if it's not reparable, you also have an identification pathway where the body says the DNA here is irreparably damaged, we need to kill this cell, right? So all of this is happening on a daily basis, minute to minute, while we're having this conversation. From an epigenetic standpoint, DNA methylation is a way that the body changes the way that DNA actually is expressed and how much RNA is expressed and what it's actually expressing. Most of DNA methylation has to do with upregulation or downregulation of transcription, right? So it's heavily involved in an overexpression of proteins or cells or RNA. And then histone modification, again, has to do with folding of DNA, like I talked about a little bit before. And if the DNA is misfolded, again, it puts it at risk, one, for improper coding, but also for more potential damage and then ultimately more coding. Taking it down sort of as we're going from sort of the inside of the DNA out to the cell. So you have transcription errors. So the DNA may be fine, but your body may, while transcribing into RNA, which is ultimately then transcribed into protein, you may have a mistake there at some point, right? The body's not perfect. We like to think our bodies are perfect, but they're not, certainly. And so there may be an error where the wrong RNA base pair gets put in, or the wrong protein gets put in when they're transcribing. And the body, again, has multiple ways, sort of redundant fallback mechanisms to say, hey, wait, this is wrong. Let's destroy whatever we're making because it's wrong, and let's fix it. And again, proteomic instability is once you've made a protein, does that protein remain stable in its conformation, right? There are things called heat shock proteins that come in and actually stabilize proteins until they're expressed properly. And if you have dysregulation in your heat shock proteins or don't have heat shock proteins, then you develop this instability of the actual protein molecule that you've created, and ultimately those proteins themselves can cause downstream actions that can be quite significant. So those are all intrinsic factors. That's all happening in the cell at the cellular level. When we think about extrinsic factors in the microenvironment, that's sort of the happy buzzword now in oncology, which is the tumor microenvironment. What does that actually mean? These are sort of outside selection pressures outside of the actual tumor or within the tumor environment that are pushing the tumor in one way or another from a cellular standpoint. So pH, so just the acidity or basicness of the tumor environment, can drive selection of a set of cells. Maybe you have a cell that is more resistant to an acid environment, whereas you have another cell that is less resistant and dies more easily at a low pH. And so that will drive selection of these cells towards a more acidic pH. And so as they generate more acid, that particular set of cells will then ultimately thrive. Hypoxia has been a big area that people are starting to look at, trying to understand it, and how that drives angiogenesis, or the creation or formation of new blood vessels, right? So we know that tumors sort of hijack the body and secrete their own vascular growth factors, and they grow their own blood vessels. And the vessels tend to be leaky and not very good, but they're enough to feed the tumor and grow the tumor, right? So how does hypoxia, or the actual internal generation of low oxygen levels within the tumor, then cause an upregulation of this growth of blood vessels, right? So it's a feedback loop where the tumor actually creates a hypoxic environment, which then drives the growth of these abnormal blood vessels. There also may be a selection for resistance to hypoxia, so kind of like making a tougher tumor. In addition, there is evidence that hypoxia within the tumor will actually drive tumor cell migration. So almost you're making a tougher tumor that essentially says, oh, I can't breathe, and they go somewhere where they can, right? And so they actually will then migrate, and they may cause an anti-tumor response. They may downregulate an anti-tumor response. So there's actually a group up at Harvard that's looking at ACE inhibitors and giving ACE inhibitors along with chemotherapeutics. And the ACE inhibitors are designed, or the thought is that the ACE inhibitors are going to relax the vasculature within the tumor and allow for better penetration of different therapeutic agents and downregulate the hypoxic response of the tumor. Now, this is, I think we're pretty early in their experiments, and they've had some mixed responses, but it's kind of an interesting idea. It's actually started by a guy who was initially an engineer and went into medicine, and so he kind of thinks along those lines, which is always a cool thing. So for paracrine signaling, so this is actual signaling between cells, so cells talking to each other and cells talking to their matrix around them. So we oftentimes forget that there's this whole extracellular matrix that is fibroblasts and scar tissue and blood vessels and interstitium that's holding all these cells together. So you could take a tumor, you could strip away all the cells, and you'd still see this matrix of tissue that sort of holds it all together. And there's actual signaling between the tumor and the surrounding cells and the matrix that help drive tumor progression. So again, when we start to think about what's happening for cancer evolution, we have kind of two different ideas about how this happens. So we have a linear evolution where the subclones of the tumor sort of arise sequentially. I happen to think that's probably not the way this happens most often, but rather there's a branched evolution where you have multiple subclones, then they make a clone of themselves, then another subclone, then another subclone. So you end up with multiple populations. Now there's data to support both. I think the bulk of people would tell you that more often than not you have a branched evolution rather than a linear evolution of tumor. But and we don't yet know when that happens or have a way to figure out when it's going to happen pre-hoc. And that would be pretty powerful if we one day figure that out. Furthermore, you have something called clonal sweep, which is where one subclone emerges and sort of outcompetes the rest of the tumor population. You can think of them as plants, right? So if you have a nice, beautiful lawn and you plant a weed in the middle of your lawn and you leave it alone, 3 months later you're going to come back and your entire lawn's going to be weeds and no more grass, right? So kind of the same idea where if the grass was a single tumor type and then this one tumor cell came along that could outcompete all the other tumor cells, then it would sweep them out of the way. For incomplete clonal sweep, again, this is the idea behind this clonal heterogeneity, right? So if you have incomplete clonal sweep, you have a subtype that comes into existence that then is able to compete against the other cell types in existence, but not to the point where it wipes them out, but to the point where it's able to set its foothold and set its stake in the tumor microenvironment and then further generate subclones that will then compete against itself as well as the cells that it was competing against previously. This idea of subclonal genotyping and looking at these different subclones has now been talked about as being used as monitoring for tumor evolution over time, right? So I'm going to talk a little bit later about some of what we are doing to use this heterogeneity, and part of that is monitoring the tumor and trying to understand what's happening longitudinally. I think very much, we may never cure extensive stage cancer or advanced stage cancer, but we may very much make it a manageable disease. And we're seeing that more and more now, where it's becoming a disease that people have over years as opposed to weeks or months. Very much like HIV and AIDS, where it was a death sentence as soon as you got it, and now people live for years and years with that virus. So when we think about heterogeneity and what it means to us clinically, it's clearly a major factor that leads to therapeutic resistance, right? So if you think about what's happening, our chemotherapeutics, short of cytotoxic therapeutics and even a applied to this idea of cytotoxic chemotherapeutics. You have cells that are susceptible to your therapies, and then you have cells that are not susceptible to your therapies. And oftentimes, you have these cells that are highly susceptible and are the predominant strain, and you treat the patient, and their tumor regresses, and then 6 months later or 3 months later, all of a sudden the tumor is growing again in the face of cytotoxic chemotherapy or even targeted therapy or even immune therapy. And that clearly means that you've now subselected out for a subclone that is resistant to what you're actually giving the patient. We've now had some major advances in terms of the way that we're able to look at these cells. We've developed what has been termed deep tumor sequencing. We've also looking at single-cell RNA transcriptomics and single-cell DNA transcription. A lot of that comes from the fact that we unlocked the genome about a decade ago. We're now working on the proteome. So I think, 20 years from now, probably 20 years from now or so, we may have a full understanding of all the proteins we actually make, which I think will be the next leap forward. So maybe at the end of my career, we'll start talking about treating people on a proteomic basis. But right now, we're thinking of them from a genetic standpoint. Again, these tumors are constantly evolving, both genetically and epigenetically. This results in increased intratumor heterogeneity. Cancer therapies, again, they produce this selection pressure, right? So if you have a cancer cell that is susceptible to the treatment that you're giving, it will die off. And again, going back to this idea that when we can actually see and know a patient has cancer, they have millions and millions and potentially billions of cancer cells. So all of them are not the same at that point. We're going to kill off an entire type of tumor or tumor cell, but there will very likely be subgroups of tumor cells that will be resistant to the therapies we give, and that will then grow over time, and those are those patients that then relapse. There are also being multiple mechanisms of persistence that are generated, right? So it's not just a single mechanism by which the cell escapes our therapies. There are oftentimes multiple different pathways that the cells use. We often say cancer is smart. We wish it was not as smart as it is. So things that also influence this clonal heterogeneity is genetic drift, right? So we talk about drift in flu or influenza, where you get drift between these different subtypes of flu, and then we get these shifts, which are these pandemic strains like H1N1 that came along. Whoops. So tumor cell loss of precise DNA replication. So again, we're damaging DNA. We're having drift away from what the original tumor was, and you're getting this heterogeneity within the tumor. You can undergo sequential progressive changes, right? So you get mutation. Then you also have genetic rearrangement. You can delete a gene. All these things can happen at the same time in a tumor cell. We at this point don't, again, have a way to forecast when this is going to happen, but we're now trying to figure out when it's happened and even potentially to back or devolve tumors in the lab to try to understand the cut points at which these happen and maybe use them to our advantage. Random drift will fix a mutation, right? So you might have a drift to a mutation that doesn't stick and that actually causes the cell to die, going back to this genetic instability question. And so if you have a total number of mutations over a selection pressure, a specific selection pressure against the mutation is greater than 1, you'll have a fix of that mutation within the population of the tumor cells. This, I think, was a really nice sort of graphical schematic of what I've been talking about. So we have a patient that's diagnosed with tumor. It grows over time. It's identified. The patient is then treated, and you see the decrease in tumor burden, but not complete resolution of tumor. And this is sort of that survival period where everyone talks about and trying to extend progression-free survival, where patients are not having increasing morbidity and even mortality associated with the tumor and the burden of the tumor on them. Then you see this shift in the slope and an acceleration of the tumor burden and the growth of the tumor. Again, and the idea now is that, again, you've now selected out this group of tumor cells that no longer responds to the treatment that you were giving them, and then you have to find another treatment, a second generation, third line, fourth line agent that potentially is aimed at some of these resistance mutations that we have found. So intratumor heterogeneity and its potential clinical implications, right? So there was a study where they looked at a renal tumor and they actually biopsied it and then went back and looked at the different genetic changes that had occurred over time and actually went back and tried to understand a phylogenetic tree of the different genetic changes that had arisen within the tumor. And it's pretty striking, I think. When you look here at this phylogenetic tree, you have the initial von Hippel-Lindau mutation that then splits with a frameshift mutation, which then sort of splinters in these various and sundry heterogeneous tumor populations within a single tumor. So people started thinking about this, and how can we use that to our advantage? Because clearly it's been a huge disadvantage to us over time. So they started to think about what did this mean from a resistance standpoint? You have the splintering of the different cell lines into different subgroups within the tumor itself, and does this mean that some of these are going to be resistant to drug, and how are these multiple routes achieved, right? So are drug-resistant cells the ones that lead to the disease progression? Maybe, maybe not, right? They may be resistant to disease, but they may not actually be the ones that metastasize. They may not grow very much. They may divide very, very slowly, right? This is sort of the big question is, how do you decide which of these tumors is going to be a quote-unquote bad actor versus which is one that you can live with for a long time and sort of watch over time and figure out how to treat as the technology changes? And which of these are resistant within a single patient, which of them are resistant within a single tumor, and how does that relate to a population as a whole as we develop treatment strategies? So when you get clonal expansion and drug resistance, normally it's an acquisition of a new driver mutation. So you get a de novo, or just, it occurs out of nowhere. You have a crossing over of the genome or damage to the genome in a way that had not previously been seen in the tumor. And you get resistance after the treatment of the tumor as a whole, and you end up with a subgroup of cells that is resistant to the treatment that you had placed the patient on previously. You can also acquire new driver mutations by selection pressure. So monoclonal driver mutations would be that you have a pre-existing resistant subclone. So again, talking about having this heterogeneity within the original tumor. You treat that original tumor and you select out for a population that ultimately was already there, but probably was being outcompeted by the sensitive cell populations, and now it becomes the dominant cell type. And then it's on us to figure out how to actually treat that now dominant cell cluster. And then polyclonal, you have multiple de novo or pre-existing subclones. So again, now you've treated a patient that has a tumor. There may have been some subclones there that previously existed that were being outcompeted. You kill off all of their competition. They start to grow. But in addition to that, you have this de novo creation of a whole new line of tumor cells that then ultimately has their own profile of resistance. And how do you deal with that? So some of these examples, so EGFR has been sort of our poster child of if you're going to have a cancer, it's great to have an EGFR mutation, and that targeted therapies are well tolerated and highly effective. However, we know that somewhere 12 to 18 months after being diagnosed and started on a tyrosine kinase inhibitor for an EGFR mutation, many patients progress, and the race has been on to find the reason for progression and what mutation is driving the resistance to the first generation tyrosine kinase inhibitor. The T790M mutation has now been sort of found as the poster child for this, in that approximately 70% of patients who develop EGFR resistance have a T790M mutation. In addition, they've also started to find this mutation alongside the standard EGFR mutant, right? So now we're seeing tumors that have both the standard EGFR mutant, and we test them also for T790M at the same time. They also have T790M, and when people have looked at this, it shows that these patients have a reduced progression-free survival on first-generation TKI, which makes sense in that you give them a first-generation TKI, you select out for this T790M, which is resistant to our first-generation TKIs, and then ultimately you have to start them on a second or third-generation osimertinib TKI that will then treat that T790M mutation. There are further resistance mutations that are being identified within EGFR that go beyond T790M. So we know that once you treat someone for T790M and then they progress beyond that, we are trying to figure out what that next level of resistance mutation is, and if there is one that is held by a large percentage of patients. So another example is MET amplification has been detected in small subpopulations of EGFR mutants prior to TKI. Again, if they have a MET amplification in addition to an EGFR mutant, they will ultimately outgrow this clone that harbors this MET amplification, which is sensitive to another form of TKI, but not the same TKI that is used for EGFR. So again, we're now seeing that there are multiple subclones within these individual tumors. Another example is ALK. So EML for ALK is we'll talk about that, a little bit about what that is in a moment, but there are multiple different kinase domains, mutations, and rearrangements that have been identified. The example here that I'm giving is 2 of them have been detected in patients that have developed crizotinib resistance. So crizotinib is the TKI that has been used for the ALK rearrangement. In addition, they've been detected as with having simultaneous bypass signaling pathways. So again, other ways of resistance around the drug that we're actually giving. So it's not a single resistance, it's multiple ways that they're bypassing the way that crizotinib downregulates the signaling pathway. And so again, we're having this release of tumor because we can block one pathway, but we're not blocking the subsequent 2 or 3 other pathways that exist, and we're trying to figure out how to actually do that and quantify it. So how do we study this? And I alluded to this earlier. So we've started looking really at single cells, and in fact, we're currently talking with a couple of companies about how to take tumors and run every single cell on a single cell assay to understand what each and every cell is doing within the patient. Now, ultimately, that may not end up being the way that we go about treating patients from a clinical standpoint, but it will certainly give us a lot of information about which way to go and how and where are the high-yield ways of getting at these problems. Furthermore, we're looking at proteomic assessments. Again, the proteome is still a bit of a black box. People are trying to figure out what all we actually make. So we ended up with a third of the genes that we actually thought that we had. So clearly it means that those genes undergo major rearrangements on a daily basis, minute to minute, second to second, to make multiple different proteins from the same strand of DNA. And how do we then wrap our head around what that actually means? And so that's the next big challenge. And that's why I say 20 years, 30 years from now, maybe we'll figure it out. But we're trying to now use some of those proteomic assessments to try to figure out what's actually happening inside of tumors and within the tumors, the different cell lines. We're taking mice, and we're always playing with mice, and we're taking mice that lack immune systems. We're trying to find mice that have competent immune systems, right? Because it's much better to study a mouse with a competent immune system that you can put a humanized lung cancer into to actually study what's really happening on from an immune standpoint. If you have a mice that has no immune system and you give it cancer, you give it cancer. So it doesn't necessarily tell you what the immune system is going to do ultimately, and that's really where a lot of the study's going. Then access to patient biopsies, right? So mice can only tell us so much, but people are not mice, right? We all read of Mice and Men, and the people were not mice in that book. So the feasibility, how do you actually get at these tumors, right? So blood, we've seen a lot of advancement in blood-based tumors, again, because it's very easy to get a sample. You just take a blood stick, or you do a bone marrow biopsy. That's not so easy for the patient, but those are not difficult or dangerous ways typically of getting to disease. For solid organ malignancies, it's much trickier because they're on the inside, and you have to stab things and go through different viscous and different organs to get to them. And then we're typically using small needles. We'd rather not do large surgeries on people and cut out large chunks of them, especially if they have advanced stage cancer, because we've already shown that probably is not good for them, for their longitudinal survival or their quality of life. So we're using small needles. Are we adequately biopsying these tumors to give a true indicator of what's actually happening in them, or are we just getting a single slice? These are all things that we're dealing with on a day-to-day basis, not to mention from a research standpoint. Looking at circulating tumor cells and circulating free DNA. So again, there are a whole bunch of companies and a lot of money, like millions and millions of dollars, going into figuring out, can we use blood tests for all of this stuff? And the answer right now is no, we can't. They are sort of adjunctive tests right now, but they are clearly not ready for prime time to be the primary diagnostic mode for anything. And then Are they representative of the disease that we see? So again, we're talking about this heterogeneity within the tumor. So if we pick up a circulating tumor cell, how representative of that, of the primary tumor and all of its cell types is it, right? And I think that's something we also don't understand, which is part of why I said that it's not really ready for prime time. And then how do you acquire quote-unquote warm biopsies? Again, this idea of biopsies, and if you need to biopsy multiple different sites on a patient, again, how much morbidity does that bring to a patient who potentially is on the back end of their life based on our current ability to treat their disease process? I think I alluded to this a moment ago, basically that a single biopsy may not represent the extent of all the driving mutations that we may be dealing with, or the resistance mutations that may come across our plate. So again, how do we deal with that? A group at MD Anderson looked at 48 samples from 11 resected non-small cell tumors, and it's interesting, they again did this phylogeny of the tumors to try to understand what was actually happening, and they looked at recurrence rates and then the branching patterns, which I thought was really interesting. And they were able to show that patients that relapsed had more subclonal branch mutations than those patients without. Now that makes sense based on all the things that we just talked about, but I thought it was very elegant for them to actually show that this is sort of what the driving force behind relapse of patients is. And again, these were all early stage patients, right? They were all 2A or less, right? So all resection candidates. But again, you have this subgroup of them that relapses. And so, can we identify based on once we've resected the tumor and look at it under a microscope and do its genomic analysis, who will potentially end up being the person that relapses, right? That's an important and potentially very powerful fact. We're not ready, we're not there yet, but I think this is one of those first papers that kind of brings this idea to people that maybe even in these early stage patients, we know there's a percentage that relapse or that recur either local, regionally, or distantly, and is there a way sort of pre-hoc to figure out who that's going to be? Sorry, there was one patient that was 3A, but everybody else was 2A or higher. So again, strategies, so thinking about these phylogenetic trees and how to understand what we're targeting, there's this idea of looking at, can you get to the primary trunk of the mutation and cut it off, right? So can you cut it off right at the stalk? That's, I think, a harder row to hoe. Right now we're looking more like at high-risk subclones, right? So again, we have a tumor, it's branched multiple times, and there's probably one or two bad actors, right? So do we go after the one or two bad actors that more readily show that they're susceptible to treatment? And then, do we look at these subclones and try to understand which of them acquired which resistance pattern, and do they share resistance patterns, and can we kind of go after them as a group as opposed to individual subclones? So there are more and more studies looking at both hematologic and solid tumor malignancies and their heterogeneity. They're tracking them at a genetic level. Again, I think this is good. It's probably not perfect yet. We're at the very tip of this iceberg. And I think, the comment should be made that when I first started in this field, it was very much about what cell type you had. You have lung, you have renal, you have GI, you have breast cancer. I think within the next 5 years, we're not going to be talking so much about the organ type or organ-specific disease in most cases, and we're going to start talking about genetic level diseases, and there's going to be this blurring, and there will still be people that subspecialize into their specific organ because there will always be nuances, but we're going to start talking more and more about these, and we kind of already are, we're more and more about these tumors as specific genetic profiles. So this is just a schematic of what we might imagine will ultimately be the way that we can apply the idea of tumor heterogeneity and the technology that we have to understand what's actually happening, and then take that back to the patient and treat them according to the information that we have. When you look at this clinical application and how we take this, this is some really nice schematics about sort of the history of what's happened in lung cancer. So in the upper right-hand corner, or upper left-hand corner, I guess, it was just all lung cancer, and then it became a subtype histologically based on squamous and adeno, or combined adenosquam or large cell. And then we broke it out and looked at adenocarcinoma, we looked at squamous cell carcinoma, we looked at the drivers of the oncogenic drivers and the mutations associated with them. And we can now drug a good number of these, and we're looking for more compounds, and there's more mutations that are coming along that we can give drug to. it's pretty exciting. Squamous cell carcinoma is still a major challenge, and still not one that has a lot of targetable mutations at this point. So again, these are sort of the main players right now: MET, EGFR, HER2, ALK, and ROS1 that we have and are able to target. There's also BRAF, there's MEK, there's a few more. I'll have a list later that you can kind of look at. So EGFR, again, sort of the poster child of mutational analysis and targeted therapy. It's a transmembrane protein. We all have it. It leads to tyrosine autophosphorylation, which is basically a long way of saying it causes a lot of downstream molecular changes that lead to upregulation of DNA transcription and cell proliferation, right? So it drives basically the growth of cells. When you have a mutant that then overexpresses it, you can imagine that if you overexpress something that drives the growth of cells, it will ultimately grow and you will develop a cancer. We, however, found a mutant that is targetable, and that led to the formation or the creation of tyrosine kinase inhibition. It's also, this is one of these pulmonary and medical oncology board questions where, it was really highly prevalent in Asian patients and in non-smokers, right, with non-small cells. So these are these patients that come to us. They're typically fairly young. They have what looks like a snowstorm appearance on their chest X-ray or their CT, and they are found to have adenocarcinoma. They're a never smoker. We test them. They are very often EGFR mutant positive, and they respond to TKI therapy. I'm going to run pretty quickly through some of these trials. They've been out for a little while, but just this is to illustrate why this is important. So the iPass trial looked at gefitinib, which is a first-line EGFR tyrosine kinase inhibitor, versus carboplatin and paclitaxel. When you looked at patients receiving gefitinib, they had a progression-free survival benefit and a hazard ratio that was essentially half of those patients being treated with standard cytotoxic chemotherapy. If they were EGFR mutant negative, however, you were doing them a great disservice by giving them a tyrosine kinase inhibitor, and in fact, you were bringing about their death at a much higher rate due to basically not treating the cell type that was sensitive to the therapy you were giving. Looking at CALGB 3040 and understanding how EGFR mutant versus wild type and its effect of TKI therapy on these two things. So again, if you have a EGFR mutant and you give them a tyrosine kinase inhibitor, their progression-free survival is significantly different, and their overall survival is also significantly different. So again, understanding that having this mutant and then treating it appropriately leads to improved outcomes was an important finding back in 2009, 2010. There are now multiple phase 3 clinical trials looking at EGFR TKI versus platinum doublets that have been performed, and they all across the board show an improvement in progression-free survival for those patients that have an EGFR mutant. The overall survival numbers are a little less encouraging, and again, I think this is a comment to the amount of heterogeneity we see within these tumors and the resistance mutations that exist and have only recently been found. So that T790M mutation was not found at the same time that EGFR mutant lung cancer was found. It was found about 5 years later. So understanding that things are changing and we're finding the way that these tumors are resistant to the drugs that we've given them has continued to make a difference, but it's not the end of the story. EML4 ALK is basically a fusion product of the tyrosine kinase inhibitor, or the tyrosine kinase domain that fuses with the kynoderm microtubule-associated protein. So basically it just dysregulates the tyrosine kinase in the same, very much similar way that an EGFR mutant causes an overproduction of cells. A different molecular mechanistic pathway, but same general idea. You get this overproduction of the downstream messaging to the DNA, upregulation of transcription, and ultimately cellular proliferation. Again, this is in addition, it also causes increased cell survival. So these cells, they don't become immortal, but they live longer and they proliferate more, again, cancer. Crizotinib was what people have coined a dirty TKI. Some of you may have heard this before. It was a drug that they were actually looking at other uses for, and they realized that patients that had these mutations were actually exquisitely sensitive to them. In fact, the first crizotinib was designed to not really cross the blood-brain barrier very well because they weren't sure what it was going to do in the brain, and now they've shown it's good and it doesn't cause lots of problems. So the second generation has improved brain penetration. So when they looked at this, they showed that they had improved progression-free survival. They looked at randomized phase 3 trials, single agent second-line therapies, cytotoxic therapies, and they all showed that crizotinib was superior in treatment of patients that were ALK fusion positive. Again, same idea. I'm not going to belabor the slide because I think our time is getting a little bit short, but again, just showing that crizotinib is effective against the ALK fusion product. Same idea. Progression-free survival is improved. However, again, overall survival, they have not really shown that overall survival is dramatically improved. And again, this goes back to this idea that we have multiple subclones. We're knocking out the clones that with the drug that we have, but we're not necessarily knocking out all the clones that matter. These are some of the trials. Again, they're showing improved progression-free survival. ROS1, same story, sensitive also to crizotinib. CMET, this MET has been a little bit tougher in that there are a couple of different ways that it can drive oncogenesis. There's amplification, there's an exon 14 mutation. It was historically thought of to be associated with just a resistance profile, but we now know that MET amplification is actually also sensitive to some of the newer drugs that are becoming available. There's a couple of studies going on looking at MET amplification and some of the investigational drugs that are showing actually very good outcomes if they have a specific MET amplification. KRAS is a bad story. So KRAS makes up approximately 50% of squamous cell and about 25% of adeno. If you're going to have a mutant, so not all of them, but if you're going to have a mutant, a lot of the time it's going to be KRAS. And it's actually been shown that if you have a KRAS mutant, you're going to be resistant to pretty much all of the therapies that we have from a targeted standpoint. And in fact, they're also more resistant, it would seem, to cytotoxic chemotherapy as well. Basically, mutant KRAS is bound in GTP, which basically causes an always-on phenomenon that just upregulates, again, all of this transcription pathways and cellular proliferation. These are a bunch of studies basically saying that if you have KRAS, bad news. This is a nice laundry list of the targets that we have, the drugs that we have for those targets, and then some of the future targets that people are working on. So the future is bright. I think I don't want to send the message that we've got these targeted therapies, but they don't matter. In fact, they definitely do matter, and the next generations are being developed that will deal with some of these resistance mutations that we find, and we have more targets that are being found all the time. So the future is very bright in terms of what we're able to do. Sort of tacking left a little bit, but importantly, we're now starting to look at harnessing the immune system, right? So getting away from actually just trying to drug a cell, but rather trying to drug the body into actually attacking the tumor, right? So the biggest way that a tumor survives is it evades detection, right? It basically downregulates our response to it and makes our body say, okay, I'm not really very interested in you. You can go ahead and grow and do what you want. So we started to think about ways that we can upregulate the immune system. Now, that is sort of fraught with some peril in that you start to turn on the immune system, you may get some effects that you don't necessarily want. So you may develop some inflammatory responses that are not so good for the patient. That so far to date has not been the case to the point where we're not able to explore these pathways. So initially they looked at CTLA-4, which basically acts at the dendritic cell or antigen-presenting cell and T cell coupling phase, where the antigen-presenting cell picks up antigen of the tumor, it hands it off to the T cell and says, hey, look, this is bad. It puts them together. The T cell gets activated and says, I need to go kill anything that has this antigen on it. It goes out, finds tumor, kills it, generates an immune response. PD-L1 is found on the tumor T cell side, right? So again, if we're thinking of three different groups, we have dendritic cell or antigen-presenting cell, T cell, tumor. CTLA-4 works over here. T cell tumor is where PD-1. So PD-1 is expressed on the tumor and actually downregulates the T cell response, right? So if you have high levels of PD-1, it actually decreases. It binds with the PD-1 receptor, and it downregulates the response of the T cell, which decreases the immune response and cell death. We now have two drugs that are checkpoint inhibitors, pembrolizumab and nivolumab, that both inhibit this. There's actually a third one, adalimumab, and I think there's a fourth one coming out. There's durvalumab that just came out, that are all looking at how to bind to these receptors or ligands themselves to downregulate the downregulation, so essentially allow for upregulation of the immune response. So there's been a series of trials. Keynote 001 looked at patients with a PD-1 expression of greater than 50%. They showed an overall survival, or an overall response rate, of 45%, which is pretty darn impressive. Keynote 10 looked at pembro versus dosi in patients that had previously been treated, so second-line therapy, and again showed dramatic improvement in terms of the overall survival as well as hazard ratio. Keynote 24, this is now looking at it first line in patients with a PS of greater than 50, and again showed statistically significant improvements in progression-free survival and response rates. Interestingly, in patients that are co-EGFR mutated and have a high expression of PD-1, you actually didn't see the survival benefit from pembrolizumab, right? So we shouldn't be giving pembrolizumab to those patients that are also EGFR mutated. Rather treat the EGFR mutant first. Nivolumab, sort of the same idea, checkmate versus keynote, and nivolumab has shown very similar responses or very similar outcomes to pembrolizumab. Interestingly, it failed in a first-line comparison to docetaxel, but when they then ultimately looked at, and they weren't controlling for the amount of PD-1 that was expressed, so ultimately when they did control, they actually did potentially see a signal there. Same idea. So, what do we learn from this? So choosing the right therapy is important. I think it's still very challenging, and we're still trying to figure out what we can do with the science that we have. Tailoring therapies are equally important, and if you live in the cancer world, which most of you who are listening today probably do, this is common conversation that we have. What's their mutation type? What's their PD-1 status? What are they doing? What have we done with the tissue? How are we using the tissue? Do we have enough? All these ideas are being tossed around on a daily basis so we can treat patients appropriately. I want to thank Chad Pico and the ACCP GAIN for the use of a couple of slides, and I'll be open to any questions.
All right, Dr. Akulian, thank you so much. Just a reminder here, let me get this, and I'll go ahead and go to our next slide in a moment. But just a reminder, on Poll Everywhere, you can submit your questions there. If you didn't do this at the beginning, just go ahead and text to the number 22333, the letters UNCCN. You'll get a message back that says join, and then you can go ahead and start submitting your questions. I've got one or two while we're waiting for others to go ahead and send those in. So you talked about the shift from the organ level discussions to the genetic level discussions. What does that look like in the clinic, both for the clinician and for the patient? How does that shift start to actually materialize in terms of that? Because obviously we're very used to talking about it on an organ-based conversation.
Right. So exactly sort of what I was alluding to right at the end is that the bulk of our conversations are now, did you get enough tissue for testing? Did you send it for testing? What was found on the testing? Do we need to retest them if they are progressing, even though they tested positive or negative for a mutation previously? Has that changed over time? Okay. And so that's the conversation that's really being had now between providers and also now with patients. So saying to a patient, hey, you've got this mutant, I've got a good drug for you that works for, we think, this amount of period of time, and then we think we may have another drug for you after that. So really, I think it's giving some hope that wasn't there prior.
Good. And again, please go ahead. We only have a few minutes left, but we do hope that you'll go ahead and submit questions for Dr. Akulian. You mentioned earlier on in the presentation talking about devolving a tumor. What does that look like? What is the process?
So there are actually people people have put together a couple of, when I say people, and not just at UNC, but at other institutions, researchers are looking at using CRISPR, which is a way that you can excise and replace different pieces of DNA within a cell, and actually excising mutations that have happened and looking at downstream mutations or upstream mutations that may change based on those excisions.
Okay.
So trying to actually understand that if you pull a mutation out, what does the cell become? What does it regress back to?
Okay.
Or if you add a mutation, what does it then become?
Okay.
And so actually trying to play with kind of the life cycle of what happened in the cell population within a single cell.
All right.
Yeah, it's cool stuff. Totally.
Yeah. It sounds like you're kind of moving back in time.
It's science fiction. Yeah.
Okay. Very good. Very good. Well, I know we are getting very close to the hour, and so that our audience. Oh, here we go. We've got one. We can get at least one question in. Is there any treatment that would be effective in older patients with no currently known targetable mutations, say 85 years or older in age?
So from a tumor heterogeneity and drug targeting standpoint, I'm not aware that there are a specific drug class of targeted or non-targeted therapies that are more beneficial. Now, there are some drugs that are better tolerated just by virtue of the side effect profiles, but we're talking at this point, when you give me a scenario of a patient that has no targetable mutations, you didn't comment about their PD-1 status, but we'll assume that they have no PD-1 expression or poor PD-1 expression, then we're talking again about cytotoxic chemotherapy. And the issue, I think, at that point, and I would defer to my medical oncology colleagues, but you're going to look for the best tolerated regimen. And potentially, if they fail that regimen, then you could invoke nivolumab, where there is data that PD-1 status doesn't potentially matter. But that's not ready for prime time from a first-line therapy.
Gotcha. All right. Well, thank you, and thank you to the member of our audience for sending in that question. We do want to thank all of our audience members for being here. We also want to thank the North Carolina General Assembly for their generous support of UCRF, otherwise known as the University Cancer Research Fund, and the UNC Lineberger Comprehensive Cancer Center. We want to thank Mary King, Veneranda Obore, and John Powell for all the hard work that they do to make this and all of our lectures possible. Coming up soon, January 26th, this Friday at noon, we've got a Lunch and Learn lecture, updating adolescents and young adults, supporting, and we just are missing a letter there, but supporting adolescents and young adults living with cancer, and that's with Lauren Lux. On February 7th, our RN and Allied Health lecture, Provider Burnout and Compassion Fatigue in the Clinic, with Dr. Meltzer Brody. And then on February 21st, our next Medical Surgical Oncology lecture, Immunotherapy Advances in the Treatment of Malignant Solid Tumors, with Dr. Pico, who you had just mentioned earlier. So we hope that you'll be there for some or all of those. We also hope that you'll visit our website, unccn.org. You'll find a recording of this lecture as well as over 200 other lectures that are available there, and lots of information about upcoming lectures. Thank you so much for attending, and we look forward to seeing you soon. Dr. Akulian, thank you so much for being here today. All right. Everyone have a great afternoon.
Goodbye.